I'm talking here about functionality. So what does it mean to achieve a target functionality? Typically, there are two classes of functionality when we talk about protein design. The first is what I would generally call binders. Okay, a binder is a protein that can selectively bind a user-specified target epitope. An epitope is just a part of another protein or a part of another molecule. And what do you use them for? Well, you can use them in different ways, but a typical one typical use for binders is to work as agonist or antagonist drugs. An agonist drug is just a drug that mimic an existing protein in your body. The existing protein normally activate a function. An agonist protein would be some protein that can activate that function even more than the natural counterpart. At the same time, an antagonist drug would be something that by binding to the same receptor that your protein does, prevents that protein from binding and it stops activation. So for example, an antagonist drug would be an antibody that can bind to that can bind to a protein, to a membrane receptor on a cell, and because the membrane receptor is what used to receive the signal from the outside to activate a cell for certain function, the cell stops working. While you can use protein binders as drugs, you can also use them purely as diagnostic and targeting agents. And you can use them, for example, for CAR-T technology and many other uses.